Welcome and thank you for joining me and us in this presentation of datasets at RxRx.AI. I'm James Taylor, a data scientist at Recursion Pharmaceuticals, and I'm joined today by my colleagues Katie Rose Skelley, formerly of Recursion and currently CTO at No Medicine, as well as Katie Heiser, research scientist at Recursion Pharmaceuticals. One of the key challenges we face in the CytoData community is to learn to extract as much biologically relevant information from high content images in order to perform morphological profiling. And today we will share with you 3 data sets released by Recursion during the last 2 years to aid the community in advancing these efforts. I'll begin by discussing machine learning with RxRx1. In 2019 and early 2019, Recursion had accumulated quite a large set of high content images, and we decided to ask the question how far we could push modern machine learning techniques to best extract biologically relevant information from our images of cell painting assays in ways that are robust to batch effects. So in July 2019, Recursion released RxRx1, a large data set consisting of 1,108 individual siRNA perturbations treated in 4 different cell types and imaged in 51 different experimental batches, yielding about 125,000 images and around 46 gigabytes of data. A key challenge to extracting images from extracting information from high content images is that often the channel intensity distributions for different perturbations within the same experimental batch are more similar to each other than the channel intensity distributions for a single perturbation across multiple experimental batches. And in the image here, you can see the siRNA 1 and 2 look more similar to each other within batch 1 or within batch 2 than siRNA 1 does to itself across batches 1 and 2, and similar for siRNA 2. So we asked researchers to learn to classify individual perturbations in ways that can generalize to unseen experimental batches. In doing so, we found that top results in a NeurIPS cell signal competition that we hosted were able to achieve greater than 99% accuracy in classifying siRNA in new experimental contexts.
The second data set that I'd like to talk about today is immune mapping with RxRx2. So this data set is a set of 434 different immune stimulants. They come from a whole host of different pathways, things like growth factors, toxins, interleukins, and different hormones. So they really run the gamut of things that could be stimulating the immune system. There are 6 concentrations per immune stimulant, and there are 12 replicates per concentration, so 12 technical well replicates. All of these immune stimulants are run against healthy primary cells, so HUVEC cells, and this data set was released in August of 2020. The data set here contains metadata, deep learning embeddings, and images. So the metadata is just sort of standard information about location on a plate and concentration for each perturbation. The deep learning embeddings, interestingly, are generated from a model that was trained on the data set James was just describing. So a data set that was trained on a whole bunch of previous batches and under siRNA perturbations, and then applied to these new immune stimulants in a completely new batch. And then we also, in addition to these embeddings, we have the images themselves. So these are the 6 channels of cell paint and 1024 by 1024 images per for each channel, for each perturbation and well. We were able to extract a lot of really interesting biology from these images and from these deep learning embeddings specifically. So we were able to confirm in-class expected relationships. So what that means is that we were able to see that, say, mTOR inhibitors tended to look really similar in embedding space to other mTOR inhibitors. Same with MEK inhibitors, and we were able to really recapture a lot of known biology, which is great confirmation that we're capturing our embeddings are actually working well. But we were also able to see novel pathways, so similarities in how these different immune stimulants acted and phenotypically looked within these cells. And many of those can be represented on this heat map by the off-diagonal similarities. So like things where mTOR inhibitors might cluster or look phenotypically similar to something completely outside that pathway. We were also able to identify mechanisms of action of novel soluble factors. So immune stimulants that we didn't previously really know what they did, but we could see how they clustered and how they looked relative to other known compounds and really get a good idea of what they were doing within the cells. So this is all on Recursion on RxRx.AI. This data set is RxRx2.
In this part of the talk, I'm going to tell you about the data sets that we released in conjunction with our COVID-19 screening efforts. We developed 2 independent disease models of COVID-19. The first, a SARS-CoV-2 infection of primary human renal epithelial cells. And the second, a cytokine storm model. In this model, we developed cytokine panels based on data from patients suffering from either severe cytokine storm or a healthy cytokine response to COVID-19. In both of these models, we screened for small molecules that reverted the disease phenotype back to the healthy state. The data from these screens were released as RxRx19. RxRx19 is broken into 2 parts, 19A, which is the data from the SARS-CoV-2 model, and 19B, which is the data from the cytokine storm model. These data sets, just like the other 2 that you just heard about, were released with both their metadata and deep learning embeddings on RxRx.AI. Notably, these are some of the largest data sets released by Recursion to date. Together, they comprise more than 850 gigs of data and more than 370,000 images. The 19B dataset is a traditional 6-channel cell paint assay with 1 imaging site per well, whereas the 19A dataset, the SARS-CoV-2 model, is a modified 5-channel cell paint with 4 imaging sites per well. If you tuned into the conference yesterday morning, you heard a lot more about these data from our colleague, Ben Fogelson, but I just wanted to give you some of the notable highlights. When we ran these screens, we included any compounds that we could find that were headed into clinical trials. Now that some of those clinical trials have read out, we have a good idea of how well this screen worked. So notably, compounds such as remdesivir and baricitinib showed positive results in our screen and also had positive outcomes for patients in clinical trials. Also, importantly, compounds such as lopinavir, ritonavir, and hydroxychloroquine showed negative results in our screen and did not show positive outcomes for patients in clinical trials. So this suggests that we were able to extract disease and biologically relevant information from these data sets. What we told you today is that machine learning models can be trained on high content cellular images to extract biologically relevant information from across diverse biological contexts. We showed high dimensional analysis of immune stimulants, which enabled functional mapping of the immune system and identification of novel interactions and MOAs. And finally, I told you about more than 850 gigs of data that we released from our COVID-19 screening efforts. We hope that these can be used to help accelerate the development of treatments against the current global pandemic. Again, all of these data sets can be found at RxRx.AI. So with that, we'll be joining you live to take any questions that you might have. Thank you for your time.